We are starting the previous one series that is from the chapter number 2, Enzyme. And today we guys will discuss about the enzyme structure and its chemical nature. Okay, so my this lecture is going to be very in very detail. So okay, let's start. As you know, the main characteristics of the enzyme that it is made up of protein, made up of protein. Enzyme is made up of protein, and the substance on which enzyme work on the substance on which enzyme work is known as substrate. Okay, so let's consider that this is the enzyme and this one is substrate and it will get attached to its active site. Okay, this is the active site. Okay, let me just attach a picture. Okay, so this one, this is the enzyme. Okay, and this is the enzyme. This is the active site. Active site is where active site is, wait, this is the enzyme, and this is the place where substrate attached is known as active site, and this one is the substrate. And when substrate get attached to active site, it makes a enzyme substrate complex. Okay, then the way, as I told you, that the place where substrate attaches known as active site. Okay, up then there are two types of active site. It is divided into two parts. The first one is known as binding site, and the second one is known as catalytic site. Okay, binding site. In binding site, the enzyme will bind and recognize the substrate. Okay, enzyme will bind itself and it recognize the substrate. And in catalytic site, you guys can assume from the name that what does catalytic means. In catalytic site, the catalyzation will take place. The enzyme will catalyze it and the product will move out. In active site, catalytic activity is also present in which catalyzation is taking place. And in catalytic site 2 to 12 amino acids are present in this site. Okay, ji. So we guys can take the example of this one picture. Like they can. This is the enzyme. This is the active site. These are the substrate. When when a substrate get attached to active site of enzyme, then it will make a enzyme substrate complex, and the catalyzation will take place, and enzyme will convert the substrate into two products. Okay, so this is the name of the catalyzation that took place in active site in catalytic site. Okay, so in this one we have discussed about the substrate and active site. Now we will discuss about the holo enzyme. Okay, holo enzyme is when the protein part and the non-protein part of an enzyme joins together, they form an holo enzyme. Holo enzyme is also known as complete enzyme. Okay, it is also known as complete enzyme. Up when let me draw it. Okay, when this is an enzyme, a protein part. Enzyme is made up of protein, so this is a protein part, and this one is a non-protein part. Non-protein part or protein part. Okay, when they both will combine together, so they will form holo enzyme. Okay, when protein or non-protein part will combine together, they form a holo enzyme. And this one non-protein part is known as cofactor. Okay, the protein part is enzyme and the non-protein part is cofactor. Let me write it down that protein part plus non-protein part is equal to holoenzyme or complete enzyme. Okay, protein part is enzyme and the non-protein part is cofactor is equals to holoenzyme or complete enzyme. Okay, ji. Up, let me write it down about cofactor. Cofactor is the part of non-protein part that is attached with enzyme is known as cofactor. Okay. The next term that we are having is apoenzyme. Okay. Apoenzyme is when the protein part of holoenzyme is known as the apoenzyme or apoprotein. This is similar to the holoenzyme. The protein part or the enzyme that is attached with cofactor in holoenzyme is known as apoprotein or apoenzyme. Protein part of holoenzyme is apoprotein or apoenzyme. Okay, let me just tell you the difference between cofactor and an apoenzyme. Cofactor is the non-protein part of the complete or holoenzyme, and the apoenzyme is the protein part of the complete or holoenzyme. Okay, so I'm done with the chemical and structure nature of the enzyme, and I hope that you guys are getting my points, and I hope that you guys will get back to my channel for the further topics. Thank you. This is all for today.